welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonise approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
All right, so this seems to be more of a working session, so maybe I can just show you what I have in terms of materials. So APIA asked us to outline sort of why we care and demonstrate that we do care about pre-analytics and how we've done this. So the overview is, of course, why is there or could there be synergy between APIA and PyCC? And then 3 sort of project proposals. Maybe just in case you're not fully aware of what APIA is doing. They work relatively closely with the National Histopathology and Histotechnology Society, and they developed this so-called TOPS program, which is a tissue optimization and pre-analytic standardization program, where they came up with certain features and basic requirements for these steps in the workflow, which I think is very good. This is also a continuous medical education approach, so I think there's certain numbers of CMEs that histotechs need, and that can be accomplished. And then we mentioned, here's a quote from you, Brandon, a whole lot of non-simple. I don't know if you recall this. This was 2019. And then, of course, one of the key issues is the pre-analytics part. And we just outlined to them that pre-analytics is a separate work group, and that there is a lot of activity in the field with a couple of papers, as well as that this was one of our first meetings back in November 2019. The breakout session pre-analytics came up with a certain slide deck, and I think you have all seen this, but sort of the key elements. One proposal was a survey focusing on potentially creating a white paper to ultimately develop what could be considered, certain guidelines and or control or quality control recommendations. And at the time, the idea was that this would take 12 months, but then, pandemic happened and this was never executed. But the idea of this sort of first project was that we need to standardize this. So I thought that is not really like directly a project, but it would illustrate that we thought about things that APIA has already kind of incorporated. I don't know if there are comments on this particular section. I think that was more or less prelude. The second project here was called OPERA, which stands for Optical Phantom for effective assessment of registration accuracy. This was kind of like color, I don't know, color reproducibility and assessment of whole slide imaging. This was at the time put together by Esther and myself to, I don't know, create a phantom or validate a phantom for submission to MDDT. There are a few commercially available, but they're all non-FDA reviewed. So the approach that we at the time thought would be a 3-month pilot where we would really take one of those commercially available phantoms and devise the scientific approach to say, can this become a regulatory grade tool? And the deliverables would be, and we worded this out kind of like, an MDT or a mock submission to really see whether this could create marketing potential or whether this would be actually turning into a product. And we spent quite a little bit of time to devise sort of at the time, what seemed to be a couple of month plan. Of course, that didn't really happen, but that's at least available so we could modify it. And then, we at the time wrote a budget justification to say that we need, someone to help manage this sort of 3-month program where we would take, I don't know, develop the program with appropriate scientific experts and then explore the pathway for a submission. So this, I don't know, was something that was devised sort of in and around that meeting at MDIC.
Bill, could I just ask a quick question?
Of course.
I'm not familiar with this term phantom. What can you describe a little bit what that is?
Yeah, so there's a slide phantom which is using several optical elements, color reproducibility, and sort of Ulla, do you have a copy of that? An image says more than 50 words. It's kind of like a Kodachrome slide, but it's printed on a glass slide. And let me just see if I find the...
Think of it like a calibration slide.
Yep.
Which has etchings of different lengths and whatnot. I think what Bill was talking about here is a color-based one. So it's got standardized colors to be reproduced by the scanner. Is there other resolution type targets too, Joe?
Yep.
The idea is to have a physical object to then image where the characteristics of the object are known, and then you can compare the image and what is expected and determine how good the imaging was.
And then how does that relate to an FDA submission? Would that phantom slide be considered a medical device on its own?
No.
It would be a tool. So you
Okay.
could use that, for example, to support a submission for, let's say, an AI algorithm. You would say, we use this tool to assess point reproducibility or focus or color reproducibility. And or you could, of course, validate your scanner against it.
Okay. All right. Thank you.
I have an image somewhere. I just can't find it right now, but it is used, for example, also in cartography and in certain instruments that are portrayed, for example, in cockpits, where you need a certain like point distance and contrast assessment. And so you would take a picture to confirm what that is, but you need to take some sort of like reference standard with that. The thing that's probably closest is a micrometer calibration slide that you put on a microscope to adjust like scaling and stuff. So it's from the same vendors, and they built it. There's one available in the US and one company has it in the UK. And I contacted both and they sent me some information. The slides are not that expensive, but they are all not aware either of the submission program, and number two, that would be probably helpful for some tools. But so in this project here, we thought that it may help to align some of the pre-analytic assessments and slide scanning calibrations.
Okay.
What the newest project here is sort of like, I don't know, we dubbed it yin and yang because the pre-analytics and the AI somehow have to come together. And the study design looks as following. So there's basically a 5-step design that starts with some reference materials that is sent out, that's collected and hosted by PyCC, that would then be available for an AI challenge, in this case for Gleason grading, by several vendors and or people who have an algorithm, and then revealing the data to illustrate whether the different materials have an impact on that. Now, I know some of these have been done, but I think the combination of a multi-institutional send-out capturing basically the variability between labs and AI hasn't been done. So let me just show you what that design would look like. So the first part would be that we pick 3 prostate cases and cut a number of blanks equal to the participating number of institutions plus 2, meaning 1, the first and last, to make sure that the tumor in between still shows the same grades. And we would scan those and assemble a panel of institutions that want to participate, and they would all agree, yes, this is adequate. And then the next step would be to take those blank slides and send those out to the various institutions. They would perform an H&E stain, scan it, and submit the anonymized file back to PyCC. So this is a classic, or I don't know, slight modification. It's not a ring experiment or so, but it would be individual slides that likely show the same tumor with the same grade as confirmed with first and last section to multiple institutions to capture the inter-institutional variability of H&E stains and scans. It's important that both of those are together. Then we would collect and host the data, and that is sort of then would be available as sort of a very small set of cases, and then we would ask vendors that have established their AI tools and or, doesn't have to be vendors, but it could also be, research or wherever, and then take those. And of course, this would be quite a few cases, and assign individual Gleason scores to these individual slides. So the data would basically consist of 3 cases and several vendors, but imagine that this is probably multiple on top of one another, because each institution basically contributes 3 different slides. So while, I don't know, we can of course modify the numbers, I think when we talk about statistics, that's of course too little, but it gets very complicated to, to orchestrate and what we're asking people to do. So I think the one discussion was to asking someone to stain 3 blanks and scan 3 blanks and upload 3 blanks is relatively little effort. While if the number of institutions participating would power this, and we can ultimately compare, at least, by individual cases, what do vendors have in terms of that. Now, I'm not too sure whether Gleason grading would demonstrate that all of this is completely consistent, or if one algorithm or one institution stands out. So for now, we said that would be, of course, anonymized. But the idea would be to kind of marry the pre-analytic inter-institutional variability with the various AI algorithms. And those were, roughly speaking, the proposals that we wanted to review today. And of course, if anyone has others, we can discuss them.
Yeah, Joe, I'll throw out 2 other ideas.
Yep.
For the HTT project, we're sourcing slides right now. And one element of the project that I haven't had to deal with yet, though, is to create slides that change the slide preparation in some, controlled fashion, to see how that affects the TIL scoring. This is kind of low on my priority list for the project, but we could elevate it, especially since Kim Glenman, who's on the team, is kind of in grants mode. Anyhow, it might be something that we could create. So I don't want to even list it somewhere quite yet until because I don't want to leave, especially the lab part of it. But you can imagine how that would be a nice contribution to my project, complementing, the diverse sampling of breast cancer patients. This would be just on a couple patients where there's plenty of tissue, or maybe even just one or two, where we really study the pre-analytic factors, which I don't know much about at all, right? But to demonstrate that things are reproducible. And so I'd be happy to go back to Kim and talk about it and potentially give something to you. Would you want, could we wrap it up in like a one-pager description sort of thing for you?
Yeah, sure. I mean, we have the project blueprint on the website. We can use that. That's even, I think that's a third of a page. So there's no, it's not as if we need any formal approach or so, but it would be great to have a quick sort of background and relevance. And I mean, I have that for HTT, so I don't need that. But what's being proposed, I think, is what they care about.
Yeah.
And whether that can be aligned with some AI, etc. So
Yeah, I don't want to give any promises right now on the spot. The other idea I have, just listening to, because I got nothing better to do with my Saturday, I guess, was listening to one of the digital pathology podcasts. And I heard Marilyn Bowie talk about a program run jointly between DPA and CAP, a quality assessment program for preparing slides and scanning slides. And basically each institution submits a small number of slides and their corresponding digital images. Now, really, those are the people that should be leading something here. I don't have the capacity really to take on much, but I think, and that seems like a group of people that are already invested and already with samples and might have good research questions. So I remember it was EPA and CAP, and Marilyn Bowie was the person talking about it. And EPA is one of the initial founders of PICC, and there should be an ability to pull them in on that. That's my other thought.
yeah, so I'm aware of the proficiency testing program for digital pathology. I don't know, Shannon, are you guys participating in that, or?
I don't know offhand.
This was maybe as much as a year ago, but probably in the summer is when I think I heard that podcast.
I think they're not organizing it as a formal proficiency test, but as a survey. So that
it was a yearly thing. She described it as something they do every year.
But I wonder if it's an education only versus like a graded one.
So we participated in the first one, and it was an educational effort in 2020, I believe. And I'm not sure, I would have to check the new catalog. But I mean, we all know Marilyn, so she's great. So we can probably, let me just double check. Because I think it's an improvement program.
Yep. I was looking at CAP's Q probes too to see if they had anything around like slide prep, and I didn't see anything like that.
I have to check the 22 catalog. Do you have that by any chance, Shannon? Because then,
Yeah, I'm pulling it up right now.
Nice to meet you, Shannon.
You as well. Yeah, it's under the education programs. I've got a digital slide program for Durham Path and a digital slide program in FNA. Neuropathology. So they've got quite a few digital options, it looks like, but they're educational.
Durham Path and FNA. And
I'll drop the link to the catalog in chat.
perfect. Uh-oh. Yeah, so I think the, just check this here real quick.
Yeah, searching for digital is a little rough because they have this
Yeah, they
digital
have a lot.
scope technology, but there you can see digital slide program Durham Path. It's the third one down.
Whole slide is also not easy.
Yes, I think I was looking on page 12. They had a couple of digital slide. Try digital slide program as your search term.
Here, digital slide program.
Yeah.
So this one here.
Yeah, those are the two that I was looking at. And there may be more. That was just a real quick search on my part.
Yep. Okay. Jinsung or Shannon, other ideas or suggestions or?
Sorry, try to find the unmute button. Joe, Brandon, there's a tech performance guidance from FDA for digital pathology, wholesale image review. Is that related to any of these projects, or this is purely institutional research, and we will not follow like similar standard or so?
Say the name again. Say the title.
It's the title is the Technical Performance Assessment of Digital Pathology Wholesale Imaging Devices.
Okay, and now that I know the document you're talking about, what's the question?
I'm just trying to see, will that one be helpful for some of these projects, even though we're going to not follow exactly the same standard, right? I can throw the link to this to the chat as well.
For sure. I mean, it's about the technical performance of the imaging system.
Mm-hmm.
So that might not be the focus of a lot of the projects, but about this project, pre-analytics, it certainly has, it's characterizing the imaging system, which doesn't seem to fit exactly in this project, at least, or in this area, pre-analytics.
Okay.
Do we have a working group that is on the imaging, right? Joe, what's the name of that working group? Scanning.
This is with Scott.
Yeah.
Slide scanning.
Slide scanning. So that's really the
Okay.
place that is
Thanks.
probably more appropriate.
So maybe we can add this here. Just so we have that at least the scanning system can follow. Okay. Yeah, so I mean, I'm totally happy to present sort of this whole thing to APIA. I mean, ultimately, it may require, you know, of course, contacting the various folks to make sure that these are presented sort of in a similar format. So we can definitely, you know, word that out or streamline it a bit.
Yeah, you're well, I'm in here now. That was not my goal today.
Where are you in there?
Slide 30.
Slide 30? You mean that the HTT project now suddenly has a new project? No.
Yes.
I mean, you can easily withdraw this right now.
I wasn't quite ready to be signing up for anything.
No, that's also not the intent. The intent is to collect the ideas. I mean, it may not be selected and or funded and or of, you know, general interest right now for many stakeholders, but it's certainly important. I don't know, it may, for example, be just a subset analysis, right, of the various slides that came from one or the other institution, but
yeah, kind of like a reproducibility study.
Shannon, you must think about these sort of pre-analytics and effects downstream quite a bit. Any sort of ideas there from your end, or?
Well, I guess, you know, as far as the one that caught my eye was the yin and yang project. You know, so that's assessing essentially a couple things. So it's assessing staining quality at the institutions, and then the scanning capability of the vendors that are selected, correct?
Mm-hmm.
There are some variables that aren't captured with that. For example, embedding quality, cutting quality, that's a big one. You know, if your slides have a lot of, or if your slides have a lot of, you know, tears or folds or things like that, that can have a dramatic impact on the ability to interpret. Now that's a lot more complicated to capture because you need a lot of tissue if you're going to have, you know, multiple institutions or multiple laboratories cut comparable slides. But I just wanted to point out that that study is catching some important variables, but it's not catching all potential pre-analytic variables. So that could maybe be, a phase 2 of that project, is looking at other, even further upstream from staining, pre-analytic activities.
Yeah, similarly fixation. I mean,
Yeah.
prostate is in a way relatively okay because one could imagine even taking a resection and splitting a tissue and sending around blocks. But I know that that's rarely ever done. I mean, also, some of the existing proficiency testing services, I think they send around slides rather than blocks.
Mm-hmm.
But it's a very good point.
I mean, it's doable. It's just I think the study design would be quite different from what is proposed here, which makes total sense. But I think that would be another interesting comparison, is cutting techniques, fixation, all of those things. And I don't know, maybe that's out of scope of this, because I mean, seems to really be focused on sort of the impacts to the digital imaging piece. And this is near and dear to my heart, because we cut our own slides, but as a large reference laboratory, we also get a lot of slides from other institutions, and it's not uncommon for us to have to request blocks to cut additional slides, because the slide quality we're seeing from the institution that's sending just isn't where we would want them to be. And so then we cut our own, and then stain and use those for the interpretation.
Yeah, I think the relevance, or one of the value propositions, is of course telepathology, and then being able to, let's say, run an algorithm on individual images if the slide is scanned remotely versus if someone sends you the slide and you scan it. I mean, we thought about, having the lab send their H&E in and run it on, for example, a one vendor platform, let's say an FDA approved scanner and algorithm. That would be the ultimate experiment, right? To say, is the FDA approved solution showing the same heterogeneity as the various other solutions that are out there? Now, it's not a competition, it's just sort of seeing whether the, let's call it like simulated real world variability through, these multiple branches affects that performance. Because if that is the case or is not the case, that would be very informative to say, what should we really look at when assessing devices. So I think that would trigger and could be very informative from regulatory science perspective.
Yeah, that's a good point, Joe. And you also showed one slide, you mentioned SVS file. I know some of the institutions, they use DICOM. So in that way, their system could incorporate the DICOM files to even by including, for example, the radiology images, right? Also, I don't know if that's you'd considered as well. And also by saying that it's the viewer, right? What the software to you views those images. I think I know to some institutions they have their own, some use commercial, right? That might to be considered as well.
So the one element that we thought in, like, I mean, those are great points. Once these are collected, just imagine, I mean, usually when slides are collected for challenges, they're unified, right? They're uniform, one format, one annotation. But I think one element that this would also capture is the various things, some vendors, when scanning the slide, portray the XY coordinate system bottom left, the other top right, the other have a vertical orientation of the slide, the other have a horizontal, some rotate the tab, some do not have the tab. And just to capture, it would capture sort of the heterogeneity. Yeah. Okay, so I like this. And I mean, the question is also, how do vendors or researchers perform? I mean, if someone says, look, this one institution provided, all these slides, so this would be the three cases, in a format that we can't read, they wouldn't be able to provide results, right? That would basically be I don't know, it's not a fail, it's just like a non-compatibility.
Mm-hmm.
And I think that would be very informative to see.
Yeah. Good
And
point.
I mean, it's, someone would ask me, can you stain 3 slides and scan them? I would say fine. I think we have to think about the overhead and, some of the concerns here. I would consider this a quality improvement initiative where I think it would be rather, this year needs some funding and we need some buy-in from a couple of experts to say we want to sit down and look at the first and last slide. I think we can probably manage that. And the cost for producing a few slides is doable.
So a couple other questions I have.
Mhm.
So oftentimes I think when you do a comparison, you've got one source of truth and then you compare everybody else against that source of truth. So, and this might be getting too into the weeds, but one thing to think about is, what is, sort of the definitive set of slides that we're going to compare everyone else against? Or is this just we're going to lay them all out side by side and kind of see how they compare? The other is, would you also want to have this expert panel not only compare the digitization of the slides, but also look at the glass slides themselves?
So just from a technical perspective, it would be, can we ascertain that if we measure, let's say, Gleason grade as the final outcome, that the grade observed in the first slide and in the last slide is the same, and whether the tumor hasn't completely changed morphologies and or, let's call it, territory on the slide, meaning region of interest. So I mean, we would need to select appropriately sized tumors where the experts would agree. Now, how they determine this, I mean, that's up for discussion. I don't think that was defined that well, or didn't discuss it that closely.
Well, I guess what I'm getting at is, if your expert panel is looking at a slide and they all agree, and I'm not a pathologist, so you'll have to forgive me. I only partially know what I'm talking about here. But if they look at the slide and they say, oh, this digital image looks poor, is that because of poor original staining? Is it because of an issue with the scanning itself? So would we want them to not only compare across institutions and compare across algorithms, but also compare digital to glass?
Got it. I think the idea was to just basically, if the send out blanks and scans will be collected by us, we hope that the folks that scan the slides would have some QA/QC in place, that slides that are out of bounds of whatever their quality metrics are wouldn't even be submitted. And I think the other element, as long as we have a ground truth that was sort of in the reference stage determined by the expert panel, it would be that ground truth that for 3 cases, I hope that experts can agree or select cases that they agree, would be compared against the AI results. So I don't think that, but of course the glass slides are still available, so we could add, a second component.
I see what you're saying. So
Yeah.
essentially the institutions would be self-selecting what they consider to be high quality slides to put into this pool.
Yep.
Okay.
But I don't know how this is usually done, whether, let's say standardizing the original tissue from one institution to capture the variability of stains from let's say this is, whatever, Mayo blocks sent out to 10 different institutions, and then we have, 10 different whatever centers or practices, or whether this should be from 3 institutions distributed. I'm not sure. This is probably easier to have one institution create this. Probably the histotech creating those slides is under close monitoring. But that gets to the point that you mentioned, do we want to standardize these factors, cutting quality, mounting, baking, all the, those variabilities? Because then we would really capture the staining variability.
So just devil's advocate, what is the value of having each institution follow their own staining process? Is in the industry, is there much variability between how we do H&E stains?
Yes.
Okay, all right.
So there are two common processes. One is called progressive and one is called regressive staining, which one is basically largely oversimplified, staying in the solution until it reaches the necessary staining quality. The other one is overstaining and then reducing the staining to the more appropriate. So there's many opinions about which one's better. I would say this simple approach is in some institutions with auto stainers, the one just works more reproducible, while others swear that they can get this done much more consistent over time when using, I think, the regressive staining. But that is, I think when you ask a pathologist, they're getting used to one of their stains, and if they see an H&E that's the opposite, they recognize it immediately, and they say, oh, they used the different H&E. But even within both kind of big brands, there are some nuances, like some institutions dilute eosin differently, some have multiple washout steps, and that gets into what I think this TOPS program is aiming for.
Mhm.
But as just stated, like I put everything in here to the point of microtomy and including mounting it on positive or charged slides or not.
Mhm.
But the staining itself is adding, I think, a lot of variability that, I mean, apart from, of course, all the other artifacts that are also important. But I think this idea was to capture or to focus solely on staining and scanning, because otherwise it gets a very, very complicated equation.
Yeah, no, I think that makes a lot of sense because, like, exactly, it's a lot harder to hold constant the upstream activities for the reasons we talked about. So I think this makes a lot of sense. I just wanted to kind of
No,
push at
this
it to
is
understand.
perfect. No, this is basically a focus on staining variability. But it would probably be good to ask for the protocol with the metadata. So if someone says, for example, I don't know exactly what the approval accounts for, and then saying that the AI algorithm or the folks that apply the AI algorithm would have to know what they're dealing with, right? So this would probably be part of the metadata that would have to be submitted with the scans. And let me just see if I find this, because this color phantom is I think I have just want to make sure that there's an example here. So this is the focus on color reproducibility and then size.
Okay.
And I think there's a couple of others available. And I'll show you the one with the for example, this is for target detection or, under the microscope. And then there's one for contrast, which has a lot of like lines and, some other challenging. And of course, There's one for rotation, there's one for angle, and one for like a micrometer. Nope.
So circling back to how this relates to FDA, and kind of, I appreciated your explanation, Brandon. That made it click for me. But would these be considered like almost like approved reference standards by the FDA, that if I'm submitting some sort of a digital pathology related submission, if I'm using these, that gives FDA some additional assurance that for quality and accuracy? Is that kind of the gist?
That's the gist.
Okay.
That's the basic gist. You know, there are some qualities that we are more interested in, like it has general use for more than just one company.
Mhm.
You know, probably also real tight protocol and even assessment, how you produce those results, you know, the performance results, you'd want to include that, I think. You know, for tools like this that are already in the community and are used and people know about them, not really, we're trying to shed light on tools that maybe aren't out there as pervasively.
Okay.
And that's my project, trying to create a data set. So generalizing more from beyond a calibration slide to an entire data set for validating an algorithm. So that whole range of possible tools would be part of that program called the Medical Device Development Tool Program.
And I think in radiology, some phantoms exist for calibration of CT scanners and MRIs to have like a non-human reference material. And
right, and those don't need any FDA clearance on it or anything like that, or tool status, because they're pervasive in the community.
no, I think this is, I mean, some of the reference materials would be very helpful to standardize, let's say, the scanning process so that you can eliminate that variable. Now that is tackling one item at a time. I don't know whether this would apply to any of the other pre-analytic processes or process steps. But I think for color reproducibility, that was sort of the focus. So that was like hyper focus on one element, but including creating, or let's say, a protocol to say how would we assess whether color is truly assessed in a reproducible fashion. I know some of the folks working with Brandon really care about that and have very sophisticated approaches. I mean, you know, color is very tricky when it comes to making it accurate and reproducible, like the color profiles, etc. So
right, and even to be that's their research expertise, and so it's not a surprise that they're interested in that. But also, I think more generally as a group, we'd like to correlate the color measurements to some sort of task-based with the pathologist in the loop performance, so that if we had an x-axis that changed color fidelity or color agreement or perturbed some pre-analytic variable and tracked the technical performance assessment of color against the human performance on a task, and can demonstrate that those two track each other, that the one maybe doesn't predict the number, but you know, when we see a peak in the technical performance, then we'll see a peak in the human performance as well. That goes a long way to saying, all right, let's not bother with the human performance assessment. We have a lot of comfort in the technical performance assessment.
maybe we can even combine the two projects, right? And send one of those reference slides around to have a color reproducible standard to see whether the difference is caused by the image acquisition or by the staining, right? Because
That's kind of where my mind was going as well. Yeah, I mean, I think these could be related for sure.
and
When
it
you
would
were
be
you were talking about some other elements beyond color that it could also measure, right, Joe? So I mean, you could get a lot of information actually by kind of putting both projects together.
well, one of the things when looking at the, this is old school. I think this was Bell Labs when they developed the CCD chips. The size of the diodes and the size of the element that's being portrayed. So, for example, some scanners use, I think, 4x or 10x or 10x or 20x for physical acquisition, with a 10 or 20x post-objective magnification to result in image acquisition at 100 or 200x, and that is portrayed onto a CCD chip that is then converted into whatever, physical magnification. The smaller, if I recall correctly, the smaller the field of a color is, the more the wavelength of the color being emitted is affected by the optical properties of the objective pathway. So meaning the smaller a field, the higher it is to reproduce color accurately. Because, red has, I think, a longer wavelength, and that is, the acquisition time plays a role as well. So for tiny little signals, the color under microscope to be really accurately represented requires a higher light source, higher energy, etc. And how scanners account for that is really tricky. So how these reference standard manufacturer companies account for that is by placing smaller and smaller fields of color onto that same slide or element. And that gets really tricky. So, some movie producers really rely on physical film rather than digital film for more accurate color capture, and then those films are later on digitized, because once they're captured, you can, apply the full armamentarium of digital, video and image filtering. But I think that's the principle of why there's smaller fields on that reference standard.
So
But
we need to put film projectors in all of our histology labs. Is that what you're saying?
I hope so, and we need to have good casting, All right. Any other comments, suggestions, or?
This was a very thought-stimulating conversation, so thank you, everybody.
All right, so we will probably put this out on a separate, I don't know, subsite and ask for, I don't know, maybe do a formal voting or present it just as is to the APIA, because they're the experts and they can see what aligns best with their TOPS program and other initiatives. And of course, since there's a patient perspective, I think that should be respected too. So perfect.
Great.
Thanks a lot for taking the time and discussing this. It's very helpful.
Thanks, everybody.
Thanks, Joe.
Thank you.
Shannon. Thanks, everyone.